All right, hi once again. We're going to start up chapter 5. We've actually kind of begun this in class a little bit, but we're going to talk about polymers. Polymers are the building blocks of all life on Earth. They're basically come in four varieties. There's lipids, proteins, carbohydrates, nucleic acids. These polymers have smaller building blocks called monomers, or one units that they then attach together to make these larger structures. So we'll go through these. We'll talk about where you find them in nature, how they're made, how they're used, what their function is, so on and so forth. So the term polymers basically is dealing with things that are large molecules or macromolecules, things that you can actually see, made of hundreds and thousands of atoms. We're going to start with the first one, which will be carbohydrates. Which you'll see, they're kind of similar to these guys, are examples of carbohydrates, mono, di, and polysaccharides. The basic idea behind them is that they're linked together through a series of reactions called dehydration or condensation reactions. In class, we talked about dropping a hydroxyl group, dropping a hydrogen, making water. That's how these are all put together. They're then broken through a process called hydrolysis, which we'll kind of learn more about as we go. So here's a quick description of what you're going to see here. So in here we have three different small units. Each one of these units is one monomer. We have monomer 1 stuck to monomer 2, stuck to monomer 3. Black lines represent covalent bonds. Here we have on either side of the polymer here that's been created, numbers 1, 2, 3, has a hydroxyl on one side, H on the other. What's going to happen is when you link these together, we're going to drop a hydroxyl group, drop a hydrogen, water's created, and then with this is a series of enzymes that help this actually happen or speed this process up. These two molecules will then bond together. So this purple one here will attach to purple number 3 here and create a longer polymer. The reverse is true. We want to break these apart. We add water between each one of these bonds in an effort to then break them down. At times you need more than water, maybe warm water. We had the conversation in class about pasta, for example. Pasta is a long string of monomers of glucose. If you took a raw piece of pasta, the hard pasta, dropped it in water, left it there for a few hours, it would start to soften up. The softening is the water breaking the bonds between glucose molecules. To make it happen faster, heat the water up, add energy to it, makes these reactions take place quicker. If we want to make it happen even faster than that, that's where enzymes come in, and we'll see that probably in lab 2 of the year. So once again, polymer basics. To break them apart, so here we have our polymer we made in the last slide, 1, 2, 3, 4. In order to break those apart, water is then added, and the hydrogen and the hydroxide from water is then split those bonds. Once again, it'll happen on its own. Once again, if you left a piece of pasta in some water, it'll dissolve or break down over time. If you heat the water, it happens faster. If you have an enzyme, it happens even faster. So the idea of the speed at which they break down in cold water, warm water, and with the help of enzymes in water, we'll kind of get through that as we move forward through these chapters. So carbohydrates are two pieces. There's a carbon and a hydrate. So for every one carbon, there's one water molecule. These, for the most part, are fuel sources in living things. Plants obviously make them. They're stored in the bodies of plants, and then we can extract the nutrients in those to then power the things that happen inside of our bodies through the process of cellular respiration. So there's mono, di, and polysaccharides. So here are some of the examples that you will be responsible for knowing. We've kind of gone through glucose and fructose in class. You need to know how to draw them. You need to know that they're isomers of one another. You need to know that they're structural isomers because they have the same formula but different shape. Glucose, we talked about, was a hexagonal shape, where fructose is a pentagon. They're both hexose sugars. The term hexose means 6, and it's 6 carbons in that sugar. As we move through some of these chapters, you're going to see we'll talk about pentose sugars. A pentose sugar doesn't necessarily mean that the shape is a pentagon. The pent portion means that there are only 5 carbons in that particular sugar. Two common disaccharides, or two ring sugars, are sucrose and maltose. Sucrose is a super sweet sugar found in It's table sugar, basically, found in a cane plant, sugar cane, things like that. Maltose is little bit more bitter. We talked about malted milk balls. It's a glucose plus a glucose, just a different flavor because it has two glucose molecules as opposed to a glucose and its isomer fructose. So because the shape is different, the end result of those are also different as well. Starches, we ran through this in class a little bit. Um, Starches are polysaccharides, are many sugars. They can be simple or they can be branched, something we'll get into as we move. Once again, monosaccharides, we drew these guys in class, so I'll skip this one. So a couple terms that show up here on the side, aldose and ketose. Anytime you see ose, ose always has to do with sugars. So there are aldose sugars and ketose sugars. I hope that the term ald and keto rings a bell to you. There is aldoses have a C double bond O at the end of a molecule, or aldehydes, where ketones have a C double bond O in the center or between carbons. Notice this one is between carbons. Interesting as I sit here with this video. There's this whole new keto diet out there, and it talks about putting your body in ketosis. Ironically, I don't know. I'm assuming fructose is probably the major component of many of the foods that people are worried about eating. If you think about, go to the grocery store, high fructose corn syrup is something that's in a lot of the foods that we eat. It's a derivative from corn plants. So they take the corn, they extract fructose from it, and they can create this high fructose corn syrup, which they then put in as a base of many processed foods. So I think the idea of a diet like that is to put yourself into ketosis, which is where your body does not. It's craving this fructose, but you don't give it to it. So the idea is that they don't have it stops producing enzymes and what have you to actually make you digest that. So it's kind of interesting on the side. But all right, ring formation. So when we were drawing these in class, we talked about the fact that sugars tend to form rings. For our purposes, that's if you see this on an AP exam, you see rings with hydroxyls attached to it. 99 out of 100 times it's going to be a sugar. They actually start off linear though, or they start off in a straight line. Ironically, if you look at the bottom here, if you look at the C4, C5, and C6, that looks like something that you find in fats known as glycerol. You also have a very similar molecule at the top here in 3, 2, and 1 looks very similar to glycerol. These sugars that we make and our plants make, they actually make them in groups of 3, and then those groups of 3 are attached together to make groups of 6, and then these linear structures that are made in plants then fold on themselves. When we draw this, we talked about some of the rules for drawing glucose. So it was, draw a hexagon with an O in the ring, and then number the carbons 1 through 6, and then starting with C, every C gets an O, which we've done around here. Then we get to number 5, and 5 we never put an oxygen on because the oxygen belonged to 5. So when these rings are formed, what happens is they become dehydrogenated, or they lose hydrogens. And as they lose these hydrogens, and part of it's that fat guy, skinny guy thing we talked about, a new bond then forms between oxygen and carbon, which is where that oxygen is stuck between 5 and 1. So that's one of the reasons. I know Jeff Lamb asked this question. This is one of the reasons that we don't draw an oxygen on number 5 as we're doing this. It's because as rings are formed from these linear forms, the number 5 brings its own oxygen. All right, here's maltose. Once again, we do this in class. Drop a hydroxide or drop a HO, drop an H, we get water or dehydration. Here's our new glycosidic or 1,4-glycosidic bond. Looking forward towards sucrose. Okay, sucrose here is showing us a excuse me, a 1,2 linkage here. So once again, the oxygen came from number 5 carbon, gets trapped in the ring like this. Okay. The reason why they use the number 2 here and not the 5 carbon is just because this guy could flip either way, meaning that you could count them 1, 2, 3, 4, 5, or 1, 2, 3, 4, 5. I think they tend to count. I think they tend to try to use the lowest number for where the where the OH group would be, because there was an OH here at one point right there. Okay, as opposed to calling it 6, 5, 4, 3, 2, 1. They're calling it 1, 2, 3, 4, 5, 6. So a 1, 2 linkage or a 1, 5 linkage would work for that as well. Once again, super common sugar. This is the sugar that's found in table sugar, or sugar that you cook with. All right, so starch. Starch is that next piece that we're going to talk about. These are going to be more than single or monosaccharides, more than disaccharides. They're going to be 3 or more. All right, they come in a couple different forms. They can be really complex and branched, or they can be very simple. So here's an example of a couple that are found in plants. These are the two big ones. There's amylose and amylopectin. I like to think of it as amylose is a pretty straight shooter. If you compare them as sisters, we have kind of the good sister here, amylose, who's on the straight and narrow, and she makes these 1,4 glycosidic linkages. Okay, and we get this nice chain here that's super easy to follow. Oh, it's 1,4, 1,4, 1,4. Whereas amylopectin, amylopectin, even though it's making these 1,4 linkages just like the other, occasionally we get this branching that happens. So we can have other than 1,4 linkages, so you can take one chain of glucoses that are 1,4 and one chain of glucoses that are 1,4 and connect them together to make a more complicated structure. This, if you've ever cooked a little bit, pectin is a chemical you can buy at the store, and it's basically just a complex sugar like this that you put in jelly, and it's what causes jelly to have that kind of not quite a solid, not quite a liquid consistency, otherwise known as a colloid. And so when you're making jelly, let's say strawberry jelly, you put pectin in there, and it gives it that firmness that you want. Excuse me. So here, these are ones found in plants. These are sugars that we can digest. We can digest amylose. We can digest amylopectin. We have an enzyme in the case of amylose called amylase, ASE, that helps us digest this, which we'll get into when we talk a little more about proteins. Glycogen. Glycogen is very similar to amylopectin. 1,4 linkages the whole way through. So we find the chain here. Like here's a good example of a chain right there. 1,4 linkage, 1,4 linkage, 1,4 linkage, and then that chain gets stuck to another chain with 1,4 linkages, which is stuck to a chain with more 1,4 linkages. These produce small granules that are stored inside of your liver. So glycogen. Let's say this morning I had a bagel and didn't do anything after that. I eat the bagel. Bagel is made of amylose, like this. Okay. I digest the amylose into individual glucose molecules. I then use those glucoses in my mitochondria to produce energy. If there is extra glucose around, my body wants to store it, and it stores it in my liver as glycogen. The reason why we do that is that if we run into a deprivation or a lack of glucose, we can always go to our liver and we can digest the glucose that's inside of our liver that's stored there in this glycogen form. It's a little harder to break down, which is good for us because we don't want to break this down first. We want to break the stuff down that's in our blood or digestive system first. But if we were lacking it, we can go to our liver and pull out the glycogen from there and use those glucose molecules that are part of the glycogen. So here's a glucose, here's a glucose, here's a glucose. We can then use those for an energy source when needed. All animals do this, by the way. So there's also this alpha and beta glucose. And alpha and beta glucose really is just it's the direction or how the hydroxyl groups are placed around. We've kind of drawn it where all the hydroxyls are on one side. Okay, they really flip-flop, where there's one on one side, one on the other, one on the other, so on and so forth. This flip-flopping is because of the size of the hydroxides. All right, it also is dependent upon where the OH is when the ring closes. Okay, so here's alpha glucose. Notice it has two hydroxyls side by side, and then up down, as opposed to up down on these rings that are formed. The only reason this becomes important is when we look at this next structure. So we have alpha glucose. All right, alpha glucose once again was down here as opposed to up down. When they go up down, notice what happens at the top. Here's that number 6 carbon, number 6 carbon, number 6 carbon, number 6 carbon. Beta glucose, and this is found in starch like amylose. beta glucose, for whatever reason, it organizes itself like this: 6 on the top, 6 on the bottom, 6 on the top, 6 on the bottom. Why is this important? Starch, like amylose, amylopectin, we can digest. Cellulose, excuse me, is a starch found in plant cell walls that we can't digest. Things like, corn, lettuce have what we call fiber. All right, the fiber helps kind of clean out our intestines. It also helps with certain types of blood pressures and those kind of things. But this fiber, cellulose, is not soluble or not dissolvable by humans because we don't have the enzyme cellulase, which herbivores do. Here's an example of what I'm talking about. So let me go back one more time just to show you. Notice the number 6 carbon always on top. That's on top, on the bottom, on top, on the bottom. When you get this on top, on the bottom flip of the number 6 carbons, here's what happens. Here's that long chain of glucose. There's one four bond, one four bond, one four bond, one four bond. But what's happening? Well, it's probably easy to see with the oxygen. We know the oxygen is attached to between number 5 and number 1 carbon. So there's the 6. Between number 5 and number 1, there's the 6. Between number 5 and 1, there's the 6. Between number 5 and 1, there's the 6. The flip-flopping of that number 6 carbon up or down causes these strands or fibers of cellulose to stick together. The problem with that is humans or carnivores don't have the enzymes or the tools to break this down, these bonds that are happening here. These are like kind of hydrogen bonds that are happening here. We don't have that ability. So we can't digest certain types of fiber, and the fiber tends to be what comes from plant cell walls. There are some fibers that can be broken down by humans, but cellulose is not one of those fibers that are found in plants. You find fiber. It's bran muffins, for example, or wheat has fiber in it. Some of those we can break down, some we can't. All right, the last carbon that we want to talk about is chitin. Chitin is it's a sugar. It's very, very similar to cellulose, meaning that it makes that flipped like that beta bonds or beta glucose. The difference is, though, it's got a nitrogen attached to it as opposed to a carbon in there. So this is what we find. It actually makes up the exoskeletons or skeletons of insects, spiders, crustaceans. Okay, so in addition to being for a food source or a source of energy, carbohydrates can also be structural. This is one of the few examples. You see it here being structural, and here cellulose being structural, or something that holds plants up. For example, cell walls in plants are what gives them rigidity. That's why trees are solid and woody. The wood actually comes from the cellulose molecules. The carbohydrates or this chitin that's found inside of the exoskeletons of lobsters and crabs. If you've ever had a really hard shell lobster, sometimes you have to use a hammer to break them open. So these carbohydrates, the way they're designed, they become more nonpolar, and they're not able to be dissolved in water, just like we can't dissolve or digest this in water. If you soak it long enough, cellulose will break down, but we don't have that ability to obviously do that in our bodies. All right, this is a good place to stop, and hope this helps.